Hi, Dr. Rodriguez. Sorry, video always freezes. It's Paxton Barcos. My last video for you is December 18th, and I am going to show you some of the structures that I found in my cow eye dissection, which I'm guessing you didn't want to see the whole dissection, so I hope it's okay that I'm just pointing out structures after the fact. But anyway, I'm going to bring my computer down here like this, as close as I can. Let me see if I can push my computer back a little bit so that you can see all these structures. Okay, I hope that this is good enough. I'll do my best to point everything out to you. So firstly, what I want to say about the eye. So the eye is a hollow structure that's divided into two compartments. We have an anterior chamber or cavity and a vitreous chamber or cavity. And both chambers are filled with fluid, and they're separated from each other by the lens, which is this structure right here that I'll describe in more in a second. So they're separated from each other by the lens of the eye. Now the anterior chamber, which I cut into, at the beginning of the experiment, is a fluid-filled space between the cornea and the lens. It includes the iris, and the fluid it's filled with is called the aqueous humor. And it's the aqueous humor is secreted by ciliary epithelia that supports the lens. Now you can kind of see this paper towel, this like huge wet mark. That's actually the aqueous humor that, as I cut into the anterior chamber, it ran out. It's kind of this watery plasma-like fluid. It was pretty gross, I won't lie, to cut into. Anyway, the area behind the lens that extends to the posterior part side of the inferior eyeball is also where the retina is located. This is called the vitreous chamber. Now, this chamber is filled with the vitreous humor, or also called the vitreous body, and that's a clear matrix that's jelly-like, and it maintains the shape of the eye. And I actually kept it right here. So I'm not sure how well you can see it, but it's just, I'm kind of pushing on it. It's just this jelly-like substance. Okay, so the entire eye is surrounded by this tough connective tissue called the sclera. And the sclera is the white portion of the eye that extends even in front of the lens. The sclera is made up of collagen and elastic connective tissue, and its job is to cover and protect the entire eye. So you can see some of the sclera here, and then obviously we have it on the outside here, which when I had to cut away all the tissue, it was kind of firmly attached to the sclera. Now the sclera is kept moist by a thin, clear mucus membrane, which is called conjunctiva. Okay, so now the cornea. So the cornea is right here, and it's the portion of the sclera that covers the anterior chamber in front of the lens. So when light enters the eye, it's first refracted or bent through the cornea, which is really interesting. And in this dissection, it's this cloudy blue looking structure, but I believe that when the cow was actually alive, it was actually that clear color. Okay, so now the iris. So the iris, I'm not sure how well you can see this, but the iris is right in here. It's a circular disc that's in front of the lens. That's the lens right there. And the iris controls the amount of light that enters the eye, and that's because it's attached to muscles that dilate or constrict the pupil, which is the opening. I don't know if you can see it right there, like clear little opening in the middle of the iris. So the color of your eye depends on how much of the pigment melanin you have in your iris. So the more pigment you have, the darker your eye color will be. And in this dissection, like I said, it's this black circular kind of disc right here. Okay, now the lens, which is moving around now, but the lens is behind the cornea and the iris. It's the second place where light is refracted in the eye. Now the lens is attached to ciliary muscles that can stretch or loosen, and this obviously changes the shape of the lens. Now this is important because changing the shape of the lens is going to alter the path of the light rays, which changes the focus of the light and ultimately the focus of the image that's seen at the retina in the back of the eye. So when someone has cataract surgery, the surgeon is replacing this cloudy lens with a clear plastic one. So I thought that was really interesting. And the lens is very thick and firm. It's I kind of think of it as like a nut, And I was very surprised by that. I thought it was a very kind of thin piece of, well, almost like plastic. So that was interesting. And it, you can see it's kind of this creamy, very light brown color. I believe that that's probably because the eye has been preserved for a while. Okay, so now to the retina, which is in the posterior portion of the eye. Now, the retina is a layer in the posterior area of the eye that contains photoreceptors. Now, photoreceptors are very important because they are the specialized neurons that receive light energy from the environment, and then they convert this energy into electrical signals that are sent to the brain for interpretation. So we have two types of photoreceptors. We have cone cells and we have rod cells. Cone cells detect colors. Rod cells are used for night vision. Now, in the dissection, unfortunately, most of the retina came off. It kind of, I think, when I just cut into it, but I saved as much of it as I could right here to be able to show it to you. And you can see there's all these little black specks in it. So that's the retina right there. It did fall off. But interestingly, the choroid, I hope I'm saying that correctly, is a vascular layer of the eye that's between the retina and the sclera, which remember is the white of the eye. And the choroid is important because it provides oxygen and nourishment to the retina. Now, in some animals, such as the cow, the choroid has an area that doesn't contain melanin, and this area is called the tapetum lucidum, and it's highly reflective, and basically it allows for better night vision by reflecting the light in a very controlled manner. And so the choroid is actually, I'm going to move the lens out right now, this shiny, kind of pearly blue layer right here that you can see a bit of. And if we pull this back, then we can see the sclera right there. It's interesting. Okay, last things I want to show you. I'm not sure how perfectly you'll be able to see this on video, but right here, there's this little hole or kind of circle, and this is the optic disc. Now, the optic disc is the place where the optic nerve and arteries and veins enter or exit the eye. There's no photoreceptors on the optic disc, so it basically creates an area of the eye where light's not perceived. So the optic disc is commonly referred to as the blind spot. Now, the optic nerve, I'm just going to flip this over, and I'm pulling it up right here. You might not be able to see, but it does really stick out in my dissection. I saw it literally immediately before I cut any tissue away. It's right there. I'm kind of wiggling it. Now, the optic nerve connects the eye to the brain. So basically, photoreceptors in the retina are going to receive the light energy. They're going to convert the light energy into electrical signals. Then the optic nerve right here is going to dispatch these electrical signals to the visual cortex, which is a part of the brain in the occipital lobe that interprets these electrical signals as images. So again, optic disc is right here, and then it runs through, and perfectly on the other side, you can see the optic nerve. All right, bringing this back up. All right, well, I think that's everything. I hope that I've taught you a lot about the eye. I definitely learned a lot. Thank you so much for all of your support, encouragement over the semester, and grateful to have gotten to meet you. And I look forward to keeping in touch with you in the future and letting you know what happens with nursing school. So thanks so much, Dr. Rodriguez. I'll talk to you soon.